Meiosis, a paper slide presentation by Bell and Louisa.
Meiosis is the process that produces gametes. Gametes are
A gamete is a mature haploid male or female germ cell that is able to unite with another of the opposite sex in sexual reproduction to form a zygote, a sex cell.
gametes are not to be confused with somatic cells, which are produced during mitosis. Somatic cells are
Any cell of a living organism other than the reproductive cells.
Meiosis is important because it ensures that all organisms produced via sexual reproduction contain the correct number of chromosomes. Chromosomes are
A chromosome is a thread-like structure of nucleic acids and protein found in the nucleus of most living cells, carrying genetic information in the form of genes.
Meiosis also produces genetic variation by way of the process of crossing over.
Crossing over is the exchange of genes between homologous chromosomes, resulting in a mixture of parental characteristics in offsprings.
Alright, phase 1 is interphase. In interphase, the cell gets itself ready for meiosis. Just like mitosis, the cell grows during the G1 phase, copies all of its chromosomes during the S phase, and prepares for division during the G2 phase. DNA replication occurs in this stage.
DNA replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules.
Prophase 1. In this stage, the cell has two N chromosomes, or a double chromatid. N is a haploid number of chromosomes. Haploid means
A haploid of a nucleus or a cell is having a single set of unpaired chromosomes. The haploid number in humans are 23.
this is not to be confused with the term diploid.
Diploid of a cell containing two complete sets of chromosomes, one from each parent. Humans have 46.
Continuing in the prophase 1 stage, the chromosomes condense through homologous pairing, or synapsis. Homologous pairing is
Chromosome pairs that are similar in length, gene position, and centromere location.
This also refers to the term bivalence.
Bivalents are a pair of homologous chromosomes.
And synapsis means
The synapse is the pairing of two chromosomes homologous that occurs during meiosis. It allows matching up of homologous pairs prior to their segregation and possible chromosomal crossover between them. It takes place during prophase 1.
after the chromosomes condense, the nuclear envelope breaks down. Crossing over also occurs in this stage. Metaphase 1. In this phase, the spindle microtubules move the homologous pairs to the equator of the cell. The orientation of the chromosomes on either side of the equator is random and independent of the homologous pairs.
Random orientation is the inheritance of one gene trait is independent to the inheritance of another gene trait.
Metaphase 1 is the phase that chiasmata occurs in.
Chiasmata is a point at which paired chromosomes remain in contact during the first metaphase of meiosis, and at which crossing over and exchange of genetic materials occur between the strands.
Anaphase 1. In this stage, the homologous pairs are separated. One chromosome of each pair moves to each pole. Telophase 1. In this stage, the chromosomes uncoil. There's a reduction of the chromosome number from diploid to haploid, and the chromosomes gather at the poles of the cells. Cytokinesis also occurs after this, which means the cytoplasm divides and we're left with two cells. In prophase 2, the chromosomes, which still consist of two chromatids, condense and become visible as a new spindle forms around the chromosomes. In metaphase 2, the chromosomes once again line up at the equator of the cell. In anaphase 2, the centromeres divide and the chromatids move to the opposite poles of the cells. In telophase 2, the chromatids reach opposite poles, a nuclear envelope forms around each set of chromosomes, and cytokinesis occurs, so the cytoplasm divides. The final product of meiosis are 4 haploid cells in which each chromosome has just one chromatid. In humans, the products of meiosis are sperm or egg cells.